Hi everyone, welcome back to Neuroscience for Non-Majors. This is Unit 5 and it's part 1 of the learning unit. And so learning encompasses a lot of stuff. There's like motor learning and language learning and decision making and stuff like that. And there's like a whole lot of stuff to cover, so it's gonna need 2 lectures. But yeah, let me share my screen. Okay, so in today's lecture, we'll learn about interaural timing difference in juvenile and adult owls. We'll learn about parts of the brain associated with different components of language. We'll learn about analogies between finches and humans and language learning, as well as the catecholamine and monoamine neurotransmitters. And we'll end with talking about the direct and indirect pathways of the basal ganglia. But to start, I'm going to talk about the owls. So the auditory system is used to identify kind of what a sound is and where it is coming from. And owls and humans both use interaural timing difference, or ITD, where basically a sound coming from the left will arrive sooner at the left ear than at the right ear, and vice versa, to figure out which side a sound is coming from. And owls can also use this timing difference to determine the elevation of sounds, while humans can't, because owls have their ears structured so that one ear is facing like kind of more upwards and the other ear is facing more downwards. But humans, since both of our ears are doing that, they can't really do that nearly as well. But if we place a speaker into each of the owl's ears, we can actually measure the effects of this interaural timing difference by playing sounds at different times in one ear or the other at a difference of a few dozen microseconds. And if we play the sound in the right ear a few dozen microseconds before we play it in the left ear, then the owl will look to the right, and vice versa if we play the sound in the left ear first. If we play the sound simultaneously in both ears, which is 0 ITD, then the owl will look straight ahead. Now, ITD measurements can be used to our advantage in measuring how juveniles versus adults adapt to an altered sensory perception. By putting prism goggles on a juvenile owl that shifts their visual world 20 degrees to the right, the owl will look 20 degrees to the left to see what is straight ahead of it. So if a 0 ITD sound is played, the owl will look 20 degrees to the left. If minus 65 ITD is played, which corresponds to 20 degrees closer to the right, or like also 65 microseconds sooner in the right ear, then the owl will look straight ahead to see something that is 20 degrees to the right. The juvenile owls are able to reorganize their perceptual processing of audition and vision so that the sounds match up with the shifted visual information. A 116-day owl who had prisms on from 88 days old had a minus 55 microsecond shift, so it thinks a minus 55 microsecond difference in its ears is coming from directly in front of it. Within a month of having these prisms removed, if owls had them on since they were juveniles, the owls could actually return to a typical state of perception and become, just see things normally. This shows how the prism training actually created an additional state of perception rather than overriding the original organization. So actually, like later on, an animal can switch between the 2 different states of perception that it has access to, even in spite of aging. And these are kind of 2 different maps that can both be utilized. So like the juvenile training caused a second map to be present that could then be reactivated later by removing the prisms. Now, adult brains have less synaptic plasticity, and but that doesn't mean they can't also develop these different like map states. But they just need incremental changes over long periods of time in order to alter their perception to the same extent as the juveniles. They're unable to adapt to a 20-degree shift immediately. However, by using, for example, like a 6-degree shift for 20 days, then moving it up to 11 degrees for 40 days, and then 17 degrees, etc., the older owls are actually able to eventually reach that same 20-degree shift in their perception that the juveniles were able to reach. It just took them longer. And after they've completed the incremental training, the old owls can revert to normal vision just like the juvenile owls. So after reverting to the normal vision, the kind of all at once single step change works on the old owls, and they're able to switch between the two states as easily as the juveniles. So adults that have undergone the incremental training have successfully reorganized their brain circuitry to switch between the two states of perception. So that should be like encouraging that like even if you grow older, you can still, you know, learn things to the same magnitude as younger people, but it's just going to take longer. Now, language is something we also should talk about. And in order to be generative or able to create kind of infinite different possibilities, language needs to be communicative, it needs to be structured and dynamic and symbolic. And other species do have like bits and pieces of a full language. For example, a bee's dance angle can symbolize the direction of food, while the length of the dance can symbolize distance. A vervet monkey has different calls for different kinds of predators and can come up with new calls for new predators. In both of these cases, though, while the language is communicative and symbolic, it's not properly structured enough to give rise to an ability to convey complex thoughts. So our grammar and our phonemes allow us to convey complex thoughts. Phonemes are individual sounds that can be combined to affect meaning, like pig versus big. Different languages have different rules about combining phonemes and can interpret the same set of phonemes differently depending on the language. Since not all of the languages use all of the available phonemes, babies actually isolate and begin to only recognize phonemes in their linguistic environments within the first several months of their lives in a process called perceptual narrowing, which also applies to faces. And we'll get to that in the next class, because perceptual narrowing, there are good things to it and then there are bad things to it. It's a very like crucial time of development. And yeah. So anyway, semantics now is the meaning of individual words, while syntax is the arrangement of those words according to rules. Semantic processing occurs in a part of the brain called Wernicke's area, while syntactic processing occurs in a part of the brain called Broca's area. By stringing phonemes into words and words into sentences according to syntactic rules, there can be infinite grammatically correct sentences. In English, we add modifiers to nouns or subjects and to verbs, actions. The modifiers being like an adjective for a subject or another noun for a verb, or clauses in sentences. We need these syntactic rules because we do not have all of the infinite possible sentences stored in our only like 10 to the power 13 synapses, you know? You can't store infinity in that. So patients with the damaged Broca's areas are able to process word meanings fine and can understand very simple syntax. However, they're unable to understand complex syntax. For example, in the sentence, Sally was killed by Joe, they would confuse Sally with Joe, but they would be able to understand Joe killed Sally. So we need to also hold prior knowledge in order to accurately decipher the meaning of the sentence. Um, and like, because sentences can have multiple meanings, but we need to be able to apply the meaning that makes the most logical sense based on prior knowledge of the same or of similar situations. For example, like in the case of the sentence "judge rules on nude beach," most likely it's about a judge who's like just making a ruling on nude, like about nude beaches, and not about a judge at a nude beach making rulings on various matters. So when we speak, we aren't articulating words as they would actually be written. For example, the sentence, "I don't want to go to the baker's house," could sound more like, "I'd own Tuan to go to the baker's house," right? And when hearing one part of those sounds out of context, we're unable to understand it. So this is why we combine visual information by looking at like the person's mouth as they speak with auditory information in order to make an accurate prediction on what is being said, because we cannot rely on imperfect phonemes. Okay, now while bees and vervet monkeys might not have syntactic structure in their communication, many other species do. Finches, for example, string syllables together into a song. Different finch species have different syllables so that finches are able to isolate songs of their species, which, their species has their potential mates. They're able to isolate those songs from songs of other finch species. And diseased finches have badly articulated songs, and this allows female finches to kind of easily pick out the male finch mates who are healthy and can pass on healthy genes to offspring, because the healthy finches actually have better quality songs. And similarly to humans, finches actually learn their songs from other finch teachers. And they also have a babbling phase, like human babies do, as they develop their song. Finches also have analogous areas to our brocas, as well as as well as to a set of structures in our brain called the basal ganglia, which we'll talk about, and to motor cortex. All of these components that are needed for creating, and producing what's actually the song. So in the finch, during singing, the HVC, which is analogous or equivalent to brocas, communicates with area RA, which is kind of analogous to motor cortex. When learning a new song, the HVC communicates with area X, which is analogous to the basal ganglia, which then communicates with the thalamus, and that finally communicates back to HVC, as well as to other cortical areas. If the finch HVC is damaged, then a young finch's song actually becomes nonsense, and an adult finch's song also becomes nonsense. And damage to area X, which is analogous to the basal ganglia, in a young finch who has only partially developed their song, causes the finch to stay stuck with the juvenile song forever. But damage to area X, again, the finch basal ganglia, in adults does not affect the fully formed song, suggesting, again, that the basal ganglia is crucial for learning, but not so crucial for retention. Yeah, so all of this is similar to humans, where damage to Broca's area causes difficulty in producing speech in both adults and children, and then damage to the basal ganglia causes difficulty learning like new pronunciations, but doesn't affect what has already been learned. While the finch area X is approximately like the basal ganglia in terms of genetics, and the finch area RA is approximately like the primary motor cortex in terms of genetics, the finch HVC differs from Broca's area a lot in terms of genetics. This might be because birds don't have complicated syntax, which Broca's area is critical for. Also, a genetic factor another genetic factor we can consider is the gene FOXP2. So all vertebrates have the gene FOXP2, and it's highly expressed in the basal ganglia of humans. And again, the basal ganglia is involved in learning new habits and movements, though it's less important for retaining those habits and movements if they've already been learned. So FOXP2 is a transcription factor that that basically increases or decreases expression of particular genes. And we learned about, transcription, translation in the other lecture, a couple lectures ago, I think, where the RNA reads the genetic information off the DNA in the transcription process, and then it carries it to a ribosome where other RNA molecules that are carrying amino acids also arrive to the ribosome and string those amino acids together, and the amino acids release and create a protein. But transcription is it's when the RNA is transcribing the genetic information from the DNA. FOXP2 is a transcription factor which can increase or decrease that process for certain genes. Now, I have to say that the genes that FOXP2 regulates the expression of, those genes are still being researched, and we don't know yet what those genes are, and it's it's an unknown area. But yeah, so anyway, FOXP2 is a gene that's vital for language in humans, and mutations in it actually cause significant challenges with language production and comprehension. While FOXP2 is identical in most mammals, there are two mutations that only exist or existed in humans and Neanderthals. So humans might be the only species that can combine symbolic words with a very structured grammar to convey very complex thoughts and ideas and information on Earth. So now we can talk more about the basal ganglia. So the basal ganglia is a set of structures in the brain that are crucial for learning rate, decision-making, and motor sequence execution. There are two main pathways that go through the basal ganglia: the indirect pathway and the direct pathway. Now, some time ago, and this has changed, but it's helpful for beginning to understand the pathways, it was thought that the indirect pathway prevents movement, while the direct pathway encourages movement. So in the indirect pathway, the first thing to happen is lots of areas of cortex. And now I said motor cortex in this diagram is the first thing to send inputs, but the truth is that all sorts of parts of the brain are actually sending inputs to the striatum, from like memory areas, emotion areas, all sorts of sensory areas, But anyway, all these places are sending inputs to the this part of the basal ganglia called the striatum, and it's exciting this part of the striatum with the excitatory neurotransmitter glutamate. And the excited striatal neurons connect to other neurons in the striatum, which release the inhibitory neurotransmitter, GABA, onto another part of the basal ganglia called the globus pallidus external. Now, normally when the globus pallidus external is not being inhibited, its job is to prevent activity in the subthalamic nucleus. And the subthalamic nucleus normally really wants to excite this part of the brain called the globus pallidus internal. But normally, it's not able to do that because the globus pallidus external is usually sending inhibitory projections to the subthalamic nucleus, preventing it from sending anything to GPI. But when the indirect pathway is active, suddenly the globus pallidus external is being inhibited, so it's not able to inhibit the subthalamic nucleus anymore, and the subthalamic nucleus is now free to excite the globus pallidus internal as much as it wants. So, if the Yeah, so if the inhibitory GABA is being sent to the globus pallidus external, GPE, then GPE is no longer able to stop the subthalamic nucleus from getting excited, and the subthalamic nucleus is free to excite the globus pallidus internal. So the globus pallidus internal, GPI, is responsible for inhibiting the thalamus. And the thalamus is kind of the gate, or like it's the sort of gate for the motor cortex. And it's super important for exciting motor cortex and telling it to send excitatory signals to motor neurons, which connect to muscle cells and, contract or the muscles. So if the globus pallidus internal is excited, then it will super inhibit the thalamus and prevent those like move signals from being sent to the motor cortex, consequently preventing movement. All right, now there's a simultaneous checking mechanism going on over here. But before we get to that, we have to talk about the neurotransmitter dopamine and how it interacts with dopamine receptors. And if we're discussing dopamine, we might as well talk about the family of chemicals it comes from, which are, monoamines and catecholamines. So the atom nitrogen likes to form 3 bonds with atoms surrounding it. If these 3 bonds include either 1, 2, or 3 carbons, with the non-carbon bonded atoms being hydrogens, you can call it an amine group. Now, if a molecule has one of these amine groups, it's considered a monoamine. Again, if it only has one of the amine groups, it's a monoamine. The atom carbon now likes to form 4 bonds with atoms surrounding it. And a benzene group is a ring of 6 carbons with each carbon bonded to the 2 carbons next to it in either a double or a single bond, as well as to a hydrogen. Now, if you replace, now in a benzene group over here, these 2 would also just be hydrogens, like no oxygens. But in a catechol group, what you do is you replace 2 of the hydrogens with hydroxyl groups instead. And hydroxyl groups are these oxygens bonded to hydrogens. So if you have like a benzene, and you take 2 of the hydrogens out and replace them with these oxygens bonded to hydrogens, or these hydroxyl groups, then you have something called a catechol. Now, if you then have a molecule with both a catechol group and an amine group, then you get a catecholamine. So like if you take another one of those hydrogens off and replace it with something that's attached to an amine, that's how you would get a catecholamine, to clarify, because the carbons like to only form 4 bonds. And so serotonin is a neurotransmitter molecule that has that single amine group, but not a catechol group. So it is just a monoamine, but not a catecholamine. But dopamine and norepinephrine, are also known as noradrenaline, as well as epinephrine, also known as adrenaline, are 3 neurotransmitter molecules that have a single amine in them, but that amine group is also attached to a catechol group. So they're monoamines, but they're also catecholamines. So we'll talk more about norepinephrine, epinephrine, and serotonin in future lectures, but for now, let's focus on the catecholamine called dopamine. I'll just briefly also discuss how to read a sort of diagram like this, where like you're looking at this, you're like, what are these vertices? Why is nothing labeled? Well, this is like a conventional drawing that you see in chemistry, where basically the way to read it is all of these vertices are carbons, and if you see less than 4 lines coming out of a vertex, it doesn't mean that the carbon is bonding to less than 4 atoms, what it means is that it's actually, there are two more bonds here coming from the carbon, but both of those are to hydrogens. For some reason in the convention, it's that you don't write down the hydrogens because that would just get messy. So over here, this carbon is bonded to this carbon. It's bonded in a double bond to this carbon, and it also has a hydrogen. Yeah, but then you also do label the non-carbon atoms, and if there are hydrogens bonded to the non-carbon atoms, then you have to label those. But yeah, that's just how to read this kind of drawing. Yeah, but dopamine basically binds to two kinds of metabotropic receptors, and we learned about metabotropic receptors like quite a few classes ago. But D1 receptors and D2 receptors. So if dopamine binds to D1 receptors, then the D1 receptor will release proteins that kind of indirectly open sodium channels and increase the excitability of the neuron. If the dopamine binds to D2 receptors, then the D2 receptor will release proteins that indirectly open potassium channels and decrease the excitability of the neuron. So, okay, now let's get back to the checking mechanism of the indirect pathway over here. So the subthalamic nucleus is not being inhibited by globus pallidus external anymore, yeah, but it is not just sending glutamate to GPI. The subthalamic nucleus is also sending glutamate to this part of the brain called the substantia nigra pars compacta, or SNC. And now SNC produces the neurotransmitter dopamine, and it sends dopamine all the way back to those excitatory striatal neurons that motor cortex and other parts of cortex were exciting in the first place to start this whole thing. And now these striatal neurons are actually expressing D2 dopamine receptors, which are the ones that decrease the neuron's excitability. So when dopamine binds to the D2 receptors on these excitatory striatal neurons, they decrease the excitability of those neurons, making them less able to excite the GABAergic striatal neurons they connect to, which means less GABA ultimately goes to GPE. However, this checking mechanism is being modulated itself by an inhibitory projection that goes back from SNC to the subthalamic nucleus. So by giving weights, different weights to different parts of the circuit, like thinking about varying the amounts of neurotransmitter released at each junction, we get a large range of control over the indirect pathway and over inhibition of the thalamus. I should also say, just to clarify, that each of these boxes represents kind of a neural ensemble, or a group of neurons that releases this neurotransmitter represented by the arrows. Okay, now I can explain the direct pathway. So the direct pathway is a lot easier. So lots of areas of cortex, including motor cortex, excite inhibitory GABA releasing, GABAergic striatal neurons. And these neurons in the striatum, which are inhibitory, they are now able to send their GABA directly. This is why it's called the direct pathway. They send it directly to GPI. In the indirect pathway, you have to go through all of these steps in order to get to GPI, which is why it's the indirect pathway. Over here, you just need it's like a direct inhibition of GPI. And again, the indirect pathway excites GPI. Direct pathway inhibits GPI. Yeah, so these striatal neurons send their GABA directly to the globus pallidus internal, inhibiting the globus pallidus internal, which then stops the inhibition of the thalamus and allows the thalamus to send all of its excitatory move signals to the motor cortex, which then again excites motor neurons in the spinal cord, which then excite the muscle cells. And if the subthalamic nucleus is active from other inputs from the other parts of the brain, while the direct pathway is active, then its excitatory glutamatergic connections to SNC will cause, or substantia nigra pars compacta, SNC, will cause SNC to release dopamine to the striatum, like before, but this time in the direct pathway, the dopamine binds to D1 receptors on the GABAergic striatal neurons, as opposed to D2 receptors on the glutamatergic striatal neurons. And so when dopamine binds to D1 receptors, remember that the neuron's excitability is increased. So these inhibitory striatal cells are being excited with dopamine and are able to inhibit GPI a lot more, allowing for more movement. However, this is also checked by that GABAergic projection that goes back to the subthalamic nucleus and stops the subthalamic nucleus from exciting SNC. So from this, we know why signals being sent to the striatum are super important, as are the indirect and direct pathway activation patterns and degrees and dopamine. But the idea of indirect pathway will make you slow down and direct pathway will make you speed up is actually dated. And if you think about it, everything you do is translated into motor sequences or suppression of those motor sequences, from speaking to eating to whatever instinctive decisions you make. And so in a paper we will learn about next week and break down next class, we will learn that the pathways of the basal ganglia do more than you might think. They might actually enhance or suppress your intentions at any given moment in time. So next week, when we finish up this unit, we will also briefly cover today's processes in the context of infant and child development, and we will also revisit synaptic plasticity and learn about rate codes and time codes of neurons and Bayesian inference and what processes might form our implicit biases, as well as how those biases left untreated can be very detrimental to our species as a whole. And that's it for this class. And your homework for this week is, it looks a bit dense, but basically it's to think through the following situation. So it's the future and an intergalactic war is taking place. Half of the cyborg society has approached you about designing an implant that will allow them to instantly learn new super complicated motor sequences to help them dodge the attack tactics and fighting styles of the rival human and alien groups. The other half of the cyborg society has approached you about designing a translation mechanism that will allow them to learn and understand the language of the aliens faster to try and create some sort of truce. Thinking back to the circuitry of the basal ganglia, the FOXP2 gene, Broca's, Wernicke's area, the finches, the owls, and everything we've learned, choose a side of the intergalactic cyborg war and fulfill one of their requests. What is the design you come up with? What parts of the neurobiological circuitry will be enhanced? What parts will be suppressed? And yeah, that's it. These are the references. Oh my gosh, like I'm so thankful for all of these classes that I took, because I'm compiling information from a lot of them for you. So yeah, that's it, and we'll see you soon.